even the decline of freshwater ecosystems and their biodiversity is even higher than terrestrial or marine systems, but you almost don't see any examples of these trends in maps and at global scale. So that was one of the motivations to propose this symposium, that we can sit together and see what new data, what tools are available out there, and also learn from some applications of freshwater biodiversity research. So we have 2 sessions. This is the first one, which will be more focused on data, tools, environmental data, hydrological data, but also biodiversity data related to freshwater systems. And in the second session, the focus is more on applications. So it's going to be very interesting. We have at least, I think, 14 talks, and I hope you stay and join the whole sessions. And on Thursday, we also organize a workshop where we aim to go through some examples where we can mobilize the data that you're going to see today and some of the tools to manipulate, process, and apply some of these data. So the first presentation is by me. It's called Environment 90 Meters. It's a new database. It's a globally standardized environmental variables database tailored for spatial freshwater biodiversity science at high resolution. But before going into details about this data set, let me tell you about another data set, which is key because it was the baseline to create the Environment 90 Meter database. And this is the hydrography 90 meter data set. So what we did here was to take the most advanced digital elevation model at 30 meter resolution, where all the terrain corrections have been applied, and run some of the well-known hydrological algorithms, and we create a series of base layers. So you can see here flow accumulation, flow direction, drainage basins, stream channels, and the subcatchments for each of these small stream channels. What was the motivation to create this new data set? So the motivation is that normally, based on some studies, headwaters contribute up to 70% of the total stream network length. But in the global stream networks currently available, all these headwaters, so the stream order 1 and 2 and 3, are normally ignored. So by doing that, we are ignoring 70% of the area and all the biodiversity in those areas. So what you see in the background is an example of higher stream orders. This is just a screenshot of a small area in Switzerland, I think. And here you see the detail of the hydrography 90 meter. So you see all the stream segments in the headwaters. So it was a huge improvement. So this data set has 1.6 million drainage basins globally, 726 million unique stream segments, and for each of these unique segments, we have their subcatchments. And we also generated 42 stream topographical variables derived from this stream network. So what you see here is just an example of the stream networks and the small subcatchments for each of these streams. And this small subcatchments is our unit of analysis for the creation of the Environment 90 Meter database. So we summarize for a list of different environmental variables, all the values within each of these small subcatchments. Why the subcatchments? So the stream network presents some challenges, yeah? So they are connected to each other. There is a routing from the stream network, which is normally not taking into account in terrestrial analysis. So these units of analysis, they follow the topographical and topological gradients in the landscape. So they take into account the longitudinal connections within the network, but also the latitudinal connections, because the subcatchment not only encompasses where the water is, but all the surroundings. So this could be considered the lateral connection. And for biogeographical analysis and biodiversity analysis, we thought this is the right unit of analysis. So initially, we took some of the most common variables used for biogeographical analysis. So we used the digital elevation model, the 42 topographical variables derived from the hydrography 90 meter. We use climate data, for example, the CHELSA, the bioclimate variables that you already know, the model flow at 1 kilometer. We also use land cover data from the European Space Agency and the soil grids database. And this is basically what we did. So for continuous data like the climate or some hydrology or the soils, we took some of, this is the subcatchment characterized in this stream segment. And what we did was to calculate a summary statistics of the values for each of these variables. In the case of categorical data like land cover, we calculated for each subcatchment the proportion of that particular land cover. And in the third case, we took the value at the stream segment, for example, the length or the stream order. And very quickly, what we did is that for each of these subcatchments, this is the stream network, and the resolution is 90 meters, but the original data sets had a coarser resolution. So what we did was just a downscaling, assigning the same value as the original, and then we calculated here the statistics and the proportions as I showed before. And the results of these are terabytes and terabytes of tables. These are basically tables where each row is a subcatchment and the columns are the values for all these environmental parameters. So it's quite difficult to manipulate, and the way we did to make this available is by creating different platforms. One of them is the GeoFresh, where you actually can filter and download information from these tables based on user-defined point locations, but we will have the next presentation going into more detail about this platform. But you also can visit this web page, and based on this tiling of the whole world, you can go to each tile if your area of interest is in there and download the tables yourself. But we also created these R packages. It's called the hydrography R, and the workshop on Thursday will run some examples using the package, which has a list of functions to retrieve the information and help you merge the tables, subset the tables, and then use those tables for specific applications. So the paper is at this moment on review, but is open, so you can visit that link and take a look at the paper. It's in the last stage of the review process. This is work done by all the team at the Institute, IGB, and I thank you, all the supporters. Thank you. And we have 1 minute, 45 seconds for questions.
Hello, Jaime. Thank you very much for your talk. I was wondering, because it's very data intensive, but many of these environmental variables are mapped at coarser resolutions. So for applications, when you're trying to do some catchment modeling or try to use it for other applications, Is there a way to actually, like, is this nested in any way? So you can actually work with these terabytes of data?
Yeah, so the data at this moment is available only for the small subcatchments. But what we have done is to include one function in the package where you can use some of these techniques. I think the fast data is called, to cluster these small subcatchments into larger ones. And you can select the level of aggregation so that you run whatever analysis not at this detail level and not at the big basin level, but at more detail or aggregated levels as you wish. So the tables are not available for the more aggregated levels, but the functions are there to create these aggregated levels and then to process the original table. So yeah, it's quite data intensive and calculations are intensive as well. Okay, thank you. So we continue with our next speaker, Vanessa Bremerich.
Okay, thank you, Jaime. I'm Vanessa Bremerich, also from IGB in Berlin. And I'm going to present today the GeoFresh platform for integrative freshwater geospatial data processing. And as Jaime already mentioned, this is a direct follow-up on the talk before, because this platform is based on the two data sets that Jaime just presented. And it was also kind of developed a little bit in parallel. So as we had seen, just briefly recapping, there are some challenges in large-scale, high-resolution freshwater geospatial workflows. Basically, we do require these high-resolution data, but they also need to account for the inherent connectivity in these aquatic ecosystems, and they need to be kind of somehow standardized. We need this environmental information, which can provide the characterization of freshwater habitats anywhere in the world. And as you can already see or have heard, these are huge amounts of data. It creates a lot of problems with the geospatial data processing workflows and the storage and processing capacities. So just a quick reminder, we had these 1.6 million basins and more than 700 million subcatchments. And these are really terabytes and terabytes of data. Plus, we have these environmental variables that Jaime just presented. So we have something like 64 variables on hydrography, 19 bioclimatic variables, soil grid variables, and 22 land cover classes. And these are in huge tabular tables. So, of course, you can go to hydrography.org and download the whole thing and put all these terabytes of data on your own server and set up the processing workflows. But what happens if I'm only interested in a small subset of data? So, for instance, I just go to GBIF and download some occurrence points on fish species in the Bogota River here. And then I want to extract this information. And this is where the GeoFresh platform comes into play. So this platform was developed by us based on the RShiny package for the interactive user interface. And in the background, there's a huge PostgreSQL relational database that holds both the geometry information as well as the environmental tables. So the processing power comes from PostGIS for the geospatial processing and the integrated extension PG routing for all the connectivity tools. So you can access it here at geofresh.org. And now I will just briefly lead you through the typical workflow that you can find on this platform. So first, I take my occurrence data and I extract the ID column, the latitude and the longitude, store it as a CSV, and then I upload it here on in the first step, and I can already see the ID and latitude, longitude displayed. And also then I can next visualize the point coordinates, see this, check them in this little map interface. I have also different background layers that I can choose, zoom in a bit, and also overlay these small subcatchments and also the stream segments so I can check my data. Then next, this is where the processing happens. First step we always need to do is assign each point to the closest stream segment. We call this the snapping. So this point gets moved a little bit. And then for each of these points, we start the PT routing processes to calculate the upstream catchment from these points for each of them and store them in the database. And then next, you can go back to this little map window and check the points, zoom in, see if the snapping worked. Also check the new information in the table, so the new latitude and longitude, also the subcatchment ID is displayed. And if the point falls into a lake, you would also get the ID of the hydro lake. So at this point, you can go and just download this whole table, integrate it in your desktop GIS, and then also check the snapping. Next, if it really happens that a point falls into a lake, you can get some additional information on the outlet of the lake and also the individual drainage basin there. And then we go to the environmental variables. This is the environment 90M dataset that Jaime just presented. So you have these 4 categories, and you can either select the ones you are interested in or just select all of them. And then in the next step, we fetch the data from the database, both for just the small local catchment that we have here, and also aggregate it so we get the mean for the whole upstream catchment. Then if you just want to have a quick look at what you got from the database, we have this little plot window. You can select one of the variables and then just see the distribution of the local and upstream values that you got back. In the end, you would just export this whole data that you got back as a CSV, and then you can just use them in your modeling workflows, for example. So this is not finished. We are currently in the 3rd phase of development, and I will just briefly show you some things that my colleague Justiel is currently working on. So one of the things we want to integrate here is the interactive catchment delineation tool. Basically, you can click here in the map and then already get the whole catchment and the stream segments back to check where, if this is the point you were looking for example, if this is right. So it adds some more interactivity, and the same is true for the snapping. So we will add some interactive snapping tools where you can move the point to the correct location if you have some more information, for example, that this should be on the main river. And we will integrate all these processes that I just presented. We make them accessible through an API and integrate them in the hydrographer R package. So that you can more easily integrate this in larger workflows if you have more data or want to make it reproducible. And last but not least, we also want to look at barrier data, for instance, from the EMBER dataset in Europe, and add also tools for analyzing ecological connectivity. So that's it. If you have any questions, feel free to come to approach me after the session or also come to our workshop on Thursday.
We have time for one question, if there is any. There you go.
Thank you for the very interesting talk. I'm wondering if you are not, if you don't think there is limitation using R-Shiny to have scalable solution, or if you think for the operation you are making it's sufficient and it will continue to be okay.
Could you repeat what was the addition you were looking for?
The use of R-Shiny to have such a app can have a lot of limitation in terms of deployment or resources you need, maybe. So I don't know if you already seen some limitation or not for the operation.
Yes, there are definitely limitations. So this is a user-friendly tool where you can just upload your data, have a first look. But if you have huge data sets or want to have them more reproducible, then I will just lead over to Meret, who will give the next talk. So the data that is in the database is both the basis for this user interactive tool and also for the more processing-oriented workflow tools.
Thank you, Vanessa. So you have seen the data and some, let's say, tools to start manipulating this data, but there are constraints, as just mentioned. So now Meret, please. She's going to talk about the next step, which is virtual research environments as a tool to enable reproducible research workflows.
Test, okay. Okay, so how do I open my slides? Oh, okay. All right, yes, hi, I'm Meret, I'm also from IGB. Yeah, so I'm presenting this topic, but as the topics before, it's actually a group effort. So we're all working a lot together on all of these tools that we're presenting here. So I'm going to present virtual research environments. So you're going to see that there is a certain connection to the two talks before, but we're trying to take a slightly different approach in this one. I'm apologizing. I had a lot of animations in this and they got lost when I downloaded, exported and uploaded this. So some slides might look a little bit overwhelming. It wasn't supposed to be like this. So yeah, we saw before that freshwater science is, it's data-driven science, it's data-intensive science. So you need to find a lot of data, you need to access it, you need to download it, and you need to handle it in some programming language, probably R or Python in this room. And the nice thing is that a lot of tools have already been developed for freshwater analysis. For example, hydrographer, which is going to be mentioned a few times here today, I think. So especially in freshwater analysis, as you have these river connectivity, it's not always easy to analyze that kind of data. So it's not just, you know, tables that you need to somehow run some regressions on, but there is this network part that is actually not so easy to do. But yeah, there are some tools that have already been developed and that you can use on your own computer to run your analysis, but you have to install them and you're going to run into conflicting versions and into dependency problems. So I don't know who has tried to install different versions of GDAL on the same Linux server. It's not so much fun. Especially, you also need a lot of processing power and memory if you want to run big, like river network analysis. So if you don't run these little ones, but actually the bigger ones, you're going to run into some memory problems soon. But that's actually not a new problem. So lots of people have been thinking about this and have come up with new ideas. So that can be summed up in bringing your research from your laptop to some of these big ones. So actually you're bringing your your computations from your laptop to the cloud. So I like this t-shirt a lot that says there is no cloud, it's just someone else's computer. Yes, so you're moving it to some server. And that's what we're doing, and we're hoping by this to make this research more feasible in terms of computing power, memory, and all that. And to make it more comfortable as well, so you don't have the burden of like installing these things, looking for these things. And also to make the research more reproducible, because we all want our science to be open and to be reproducible and to show it to colleagues and all that. So that's also one of the goals that we have. Yeah, so I'm funded by a project called Aqua Infra. So it's funded by the European Union. I have to add this little nice line here, so thank you. Yeah, so what we're doing is we're building such a research environment mainly for freshwater researchers, also some marine stuff in there, but we're trying to focus on freshwater. So how could that look like? So we've seen in Vanessa's talk, connectivity is a big thing in freshwater analysis, and that's most easy to analyze in a graphical way. So we have this little tool where you can click on a map and you can see what's the nearest stream segments and subcatchments, what is the upstream catchment, so you click somewhere and you see the whole upstream area. You can also retrieve this whole area as a polygon and use it in your further analysis. You can also get all these little subcatchments that Jaime mentioned and get the environment 90M data for that. Yeah, and sometimes you're not interested in the upstream catchment, but you're interested in the downstream. So if I drop a rubber duck in the river, where is it going to the sea? What path is it going to take until it reaches the sea? So you can click anywhere and you're going to see the trajectory of the rivers. You can actually do all these things also from programming languages. So R, Python, whatever, but they're not as nice to show, not as fancy. So we're going back to the graphical part. Yeah, sometimes your rubber duck doesn't want to go to the sea, but it just wants to swim from one place to another. So you click on 2 points and you actually see the connection through the stream network and see how you can, or how a fish or whoever could swim from one place to the next. So all of these different kind of data results, you can retrieve them as GeoJSON and then next use them in another analysis script. Or you could call the underlying OGC API that we put on top of these tools. So OGC API, it stands for the Open Geospatial Consortium. I don't know, maybe you're familiar with that. So it's actually an HTTP API. So a request like this, a response like this. The nice thing is it works in any language. So you can do that in R, in Python, anywhere. So what it actually does is you put the processing on some server and it's super flexible. You can run whatever you want, whatever language you want. And then the client uses HTTP to request this processing and to get back the results. And this API is actually the best practice cited in the spatial data on the web best practices. So it's a widespread standard. And the way we did this is we put a platform called PyGeo API. So that's a Python implementation of the server. So that's what's kind of running the whole computations. In front of that, there is the API and the HTTP request. Now, if you don't want to do that yourself, this HTTP stuff, which is in R not so comfortable to do, you can also use a platform called Galaxy, and we've seen that before in this morning session. So Galaxy is described on its own website as an open source web-based platform for data-intensive biomedical research. And I think we can cross out the biomedical because more and more other sciences are also using this platform. So this is how it looks like. So in Galaxy you have these tools and it's a graphical user interface where you can put the parameters, click on Run Tool, and you get the results. And the nice thing is you can chain these tools into these long like workflows, and then you can actually share them with colleagues, rerun them, share them with reviewers, with students, and they can all just run them or edit them, put their own data. So that's really like the more user-friendly version. This is another example. So the Galaxy platform, so I think we may have another, like, more introduction to that maybe later. So it's a nice graphical user interface for research. You can chain the tools to these graphical workflows on the canvas. You can share workflows and data. And the nice thing is it's reproducible for anyone. And the nice thing is also there's many users and a lot of documentation, lots of tutorials, because this platform has been around for a long time. So it's not a project that's running like for a few years and then goes to the trash. So it's actually there for a long time. Yeah, to sum up, we are working on bringing the research workflows from the laptops to the servers in the Aqua Infra VRE. And we have these three approaches, this OGC, HTTP API, acronyms, the Galaxy platform for a more graphical user interface, and then various web clients, like, for example, this graphical one that I showed for more like exploratory analysis. That's it. We have 9 seconds for questions.
We are ahead of a schedule, so if there is one question from the public. Nope? No. Oh, clear. Thank you, Meret. Yes, we produce a lot of data and we have a lot of meetings talking about fair data, and it's quite challenging, and we are not sure if we are reaching that. So I'm looking forward for the next talk. It's called One Water Data Platform Towards Fair Water Data Ecosystems in France. Ivan, the floor is yours.
So good afternoon, everyone. I'm really happy to be there to present something where I don't speak too much about Galaxy for one time. But what is cool is that, in fact, other colleagues already do that. And it's really interesting to see how we can see other collaborators or people taking this kind of platform to push code to the virtual research environment. So it's really a nice example. And the project I will present now is a project who will benefit from your work, and also I hope we can also push some information, guidance and tools maybe to your project too. So I am not the leader of the project, One Water. I don't know where is, or maybe it's just me who use not the good button. So if you have questions about the project and things like that, it's maybe better to ask to the two guys who are their email at the end of the slides. I am there notably just to spread the word, but it's really a talk focusing more on fair approach related to geographical information system. I am not a specialist, but okay. First of all, I will not start by the project context, so you will see that at the end. The big issue we have classically when we want to manage data is to arrive to make something with the data, and you see that it takes time, and we have several steps to follow. And here we see that in fact we can maybe ameliorate that using fair approaches notably, and to work closely between IT guys and domain-centric guys. So why moving towards fair water ecosystems? Because when we are on the fair approach, we can ameliorate the reproducibility of research processes with something who is in crisis now. You are obliged to pay attention to not reinventing the wheel and standards using existing open source based solution, and to rationalize and mutualize technology, resource, and you can save time and money and also gain in terms of coherence. And as we are in a public monitoring state, it's also important to enable to work cross boundaries works. If we are not fair, in fact, we see that there is many barriers, and one issue we have is the silos approaches, where in fact we see that we can have issues integrating heterogeneous data or just having exchanges between silo of administrative guys, for example, and organization system. We have a risk also of misinterpretation, and so maybe uninformed decision. So we need tons of converters, translators between system silos. This is loss of time and money, often hidden cost. And we don't have virtual research environment, digital twin, shadows, AI, data space dynamically connected to the data. So how we progress throughout this FAIR ecosystem? So here, with this focus on geographical information systems vision of FAIR, where we see that we can benefit using and setting up international fair water practices, building on existing interoperability standards and practices, having support for IT solution to implement them, and deploy solution in the community, test, train, promote. So our methodology started with the production of a FAIR factory, to have a factory blueprint, notably around specification of FAIR principle. So the idea is making something related to what we call the FAIR implementation profiles. I don't know if you are aware about that, but to have manner to describe exactly what we want in our domain to implement to reach a highly degree of fairness for each FAIR FAIR principles, and documenting this and going towards concrete advices in terms of standards you can use and things like that to reach this high fairness. This allows us to have specification of what we call a fairness analysis grid, where we, for each FAIR principle, we can have clear information about what we have to do and how we can do that or not, if it's already done or not. The idea behind the FAIR factory is to support action, notably around, I will show you just notably around some example. One example is on French hydrological network through the database Carthage, where we see here a grid with field and who allow us to directly deploy a solution who is executing and using standards and solution to propose accessibility to the data here on the interoperability. But in fact, we have several examples, notably around transforming ad hoc semantics and API towards sensor things API, for example, or coverage data when it's not available through FAIR coverage API in WCS deployment, for example. And again, we can see the services we deployed behind that. With this FAIR factory, we have a FAIR water training. So training is notably done to have a joint work with OGC, WMO, and at the European level with Water for All project particularly. And in France with research infrastructure like DataTerra and the Gaia Data project, and of course with the One Water project. So the methodology also is working around FAIR data services. So to illustrate this, we have there, for example, geospatial data FAIR services, where we can request layer through WMS, WFS, or WCS protocols, thanks to GeoServer notably, or observation FAIR service, where we can request objects through sensor things protocols, using solutions like Frost or Steam, for example, or decided to have Spatio Temporal Cubes FAIR services, thanks to EDR, and notably through PyGeo API, was mentioned just before. So after that, we also need to support open source protocols, standards, and tools notably around metadata with GeoNetwork and the metadata standards oriented towards that. Our client like QGIS or MViewer, and a workflow-oriented system like Galaxy, who allow us to create virtual research environment. So to see a little bit more the FAIR factory in action, the idea is to have data and processes who are in this FAIR factory to allow us to have FAIR diagnostic. And if we need, we can go to trainings, report, sharing, to have a loop where we can understand or create our FAIR diagnostic. And then we can create catalog of this process and data, who can allow and facilitate the openness and the sharing of the content. And finally, populate virtual research environment, so the dream can become true. If we take the idea of the FAIR building blocks, you can see that you can create workflow, for example, from code to go from some data sources to derived data set you want to have. But in fact, you can combine that depending on the final object you want, so you can, etc., automatize and be rapid to create the research visualization, results, indicators, or things like that. Another example, a running example, on the TNT2 model is what we need to integrate to make this kind of modeling. When we were making that by hand, using brackets, it's something like 6 months minimum with a lot of pain to make a lot of data pre-treatment. And now, in fact, we can see that in just a few minutes, we can around that because we have FAIR protocols, FAIR services oriented and usable from catalogs, and allow us to make subsetting and having a zip file, for example, so we can easily create a visualization like here. So in conclusion, it works, there is no technical blocking element, and it's just a matter of doing and linking FAIR people together. And rapidly, the context of the project, so we have 6 applications on the One Water project, with a particular one who is on the data sharing challenges, so it's where we are working particularly and related to these talks. And the idea is to have something which is integrated with national and international initiative, both in numerical IT services, but also in data production, in data monitoring dispositif. I don't think it's English, but I try. And the idea is to create link with all international entities who are working notably around standards, thanks to the use of infrastructures nationally and at the European level. Thanks for your attention. If you have questions, don't hesitate to not ask me questions, but to just contact my colleagues. Thank you.
Is there any question for Ivan? Okay, thank you, Ivan. Very, very impressive. Paula is online, so we can ask questions if. Okay, so the next presentation is a virtual one by Paula Torres Zaffaroni. It's called Communicating Interdisciplinary Research to Aid Timely Responses Amid Emerging Environmental Risks.
Hello, good morning. My name is Paula Torres Zaffaroni from the Leibniz Institute of Freshwater Ecology and Inland Fisheries in Berlin, Germany. And today I will talk about how we can communicate interdisciplinary research to aid timely responses amid emerging environmental risk. In the summer of 2022, the Oder River, which flows through Poland and into Germany, faced a highly severe ecological crisis due to a massive plume of brackish water algae that led to the widespread fish mortality along over 90% of the river extent. This particular event took place in one of the hottest and driest summers on record in the region. And because of the large-scale consequences and the scale of the disaster, in Germany, an interdisciplinary and multi-institutional project was launched in 2023 to investigate the blooms causes, the river ecological status, and its recovery dynamics. So here on the right-hand side of the slide, you can see a very succinct representation of the structure of the Reusable project, which has 13 sub-projects, or TPs or SPs, as you may see differently, that they all tackle different aspects, going from the genomics of the algae to the growing conditions tested in the laboratory experiments, the evaluation of different levels of organization, and in the aftermath of the disaster at the floodplain level. Specifically, Sub-Project 4 aims to provide a reconstruction of the primary protesters dynamics using remote sensing-based tools. As an example, this decadal analysis of the whole Sentinel-2 archive unveiled the interannual variability of the blooms between 2016 and 2024, and the intensity of or magnitude of the blooms in the different years, as a response to differences in temperatures, discharge, and radiation. Also, in the generation of this dataset, we also aim to generate a near real-time evaluation of the river status that can help determine hotspots of algae growth and possible bloom outbreaks, even beyond the specific risks of Primesium parvum, which is the brackish water algae that I was talking before. With heat waves expected to increase both in frequency and in magnitude, and with salt inputs to the Oda River still very much unregulated, similar events like this may reoccur. So this means that we cannot only just look backwards to understand the specific 2022 bloom and its causes, but we also have to develop some tools that allow us to anticipate and to react when similar events occur or seem like they are more likely to occur and to emerge. And doing this requires integrating very different types of knowledge, going from the reconstruction from satellite imagery, which can give us near real-time information about the algae development in the waters, but also we need to integrate the very broad and sparse monitoring records that exist and that are being kept in time. And also we need to somehow integrate all the experimental work in the lab that is being created in order to understand the most relevant factors affecting Primesium parvum growing conditions and their toxicity liberation. And so the challenge here is how to integrate and communicate results across spatial, temporal, and disciplinary scales in a way that supports both scientific analysis and also decision-making, and very rapid decision-making in particular. So to tackle this, we developed a web-based data visualization dashboard to bring together a subset of the outcomes from all the Primesium-related work in the Reusable project, with specifically the inclusion of the monitoring and the reconstruction of algae activity in the catchment, the inclusion of multi-source monitoring of the most relevant environmental variables impacting or influencing Primesium growth, and also the translation of the project experiments in natural system variables. So, in other words, how do the factors that have been for the dashboard deployment, we leveraged this Sentinel-2 chlorophyll data that we hosted in a data cube and that is updated on a day-to-day basis as new images are being acquired of the river condition and the river history in blooms. So the first level is near real-time conditions. So to answer how is the river doing this week in terms of chlorophyll concentration, temperature, and discharge. The second level is past conditions, so to answer how have blooms developed and grown in the last decade. A third level of information, which is the environmental context and some results from the analysis, to answer, for instance, how do blooms increase or change in response to changes in hydrometeorological variability. This is a screenshot of, or a screen record of the dashboard and how it looks like. So first the user access an About tab to see what the project is about, what this website is about, what they can find, and how they can interact with the different elements in this website. And then we have the current status tab, which has first an overview of the chlorophyll concentration in these 1 kilometer river segments, including some relevant tributaries like the Barta River and the Nicer River. And the user can zoom in, can zoom out, and can interact with the map to check what the concentration is on each of the locations. And they can pan across the river basin, and they can also click on one of the river locations and see the 30-day traceback of the increase or decrease of both chlorophyll concentration, but also of water temperature and discharge on a much higher resolution basis, like shown here, for instance. Then the past dynamics, like I said before, it summarizes some of the analysis of the blooms detected between 2016 and 2024. And again, the user can interact with the map. They can zoom in, zoom out to see what the average concentration of chlorophyll overall has been like in each of the river locations. And also, as they click on a location, they can check the specific phenology. So when in the year and when across the years have blooms developed and with which magnitude, and in comparison also with upstream and downstream segments and so on. And the third level, the more synthesized level, that is also being under development as we test different options, is the environmental context analysis. Here, for instance, showing some of the results, some of the simple language results of how radiation regimes and temperature regimes influence the duration and the magnitude of blooms, as we have seen for the last 10 years. Currently, we are opening this dashboard for some testing with different users, and we are about to do some feedback rounds with stakeholders, specifically with project funders, with actors that are interested because they are being affected or they can be affected by such a reoccurrence, and so on. And we are also thinking for the next version that we are going to develop, how to integrate outputs from other sub-projects, specifically the inclusion of biotic community composition at different organizational levels. Lastly, we are testing how the laboratory experiments on pre-nascent growth condition constraining factors are being reflected or can be reflected in real world conditions, and how we can translate that body of knowledge that they have generated in this project in an actionable module within the near real time or the current conditions level of information. And with that, I thank you for your attention. Muchas gracias por su atención. And I think we don't have a question and answer space. So if you want to write me at my email, I would be very happy to exchange ideas or to answer any questions that there may be.
Thank you, Paula. We are ahead of schedule, so if there is a question for Paula, it might take time because she's online, but she's available if there is any. No? Okay. Thank you, Paula. So let's move on. Now we go from stream networks, data, and platforms to biodiversity a little bit. So we start with fishes and then with insects. So the next talk is by Justiel. He's going to talk about the Danube fish database. documenting species distribution across some major European river basin.
Thank you, Jaime, for the introduction. My name is Yusiel Torres Cambas. I'm from the Leibniz Institute of Freshwater Ecology and Land Fisheries, as many of us in this session. And I will be presenting a database on fish species occurrences from the Danube River Basin. This is one of the tasks of an European funded project named Danube for All, which main objective is to develop a restoration action plan for the Danube. And in this case, we have taken fishes as subjects of study. So we started collecting all of this information on fish species occurrences. So yes, to put in context the Danube, the Danube River Basin is the most international one basin globally. Actually, it comprehends 19 countries, and at the same time is the one that has the highest fish species richness, at least what is known or recorded in Europe, with about 100 or 124 species, depending on the source, and about 20% of all of those species, native ones, are threatened. And threats include, for example, the lack of connectivity, such as longitudinal connectivity and lateral connectivity, because of the presence of barriers and dams, but also the introduction of invasive species, the pollution, and all of these threats could be increased in the future due to climate change. So before this database, all the information on fish occurrences was dispersed. It was, most of them was not freely available. And also it came in a different formats. So the main goal of creating this database was to put together all of this dispersed information in a common format and making it freely available for other users. Sorry, okay. So we collected the fish occurrences from different sources. The 80% of all occurrences came from organization partners of the Danube for All projects across the whole basin, but also we take information from the different joint Danube surveys, which are surveys that are being conducted periodically in the Danube River Basin, at least 4 at this moment. Also, we took information from the European Freshwater Index and also integrated information from GBIF. To collect and format and check the quality of the data, we used R, and all the code that we used for that, we grabbed the code in a R package that we named Danube Occur. So you are seeing in the slide a screenshot of the website of this Danube Occur work package. If you scan the code, you can go, for example, to one of these, to one of the vignettes of the R package, see in more detail the workflow we follow to collect, format, and check the database. Finally, we collected more than 100 records of occurrences in 114 species. At least 83% of species have 10 or more records, and the record goes from the 19th century to 2024. in the map you can see the spatial distribution of those records. The database is organized in 36 fields, which follows the standards of the Darwin course, and those 36 fields are organized in different categories, which are summarized in the box that you see in the slides. These occurrences can be easily integrated with some of the data sets that were presented before by my colleagues, because one of the columns of the database has the subcatchment IDs of the environment 90M and hydrography 90M. So the tables from this geospatial dataset can be joined with the occurrences of the Danube fish database. This integration can be done in two ways. For example, using the GeoFresh platform presented before by Vanessa. So we can upload the points, we can snap the points to stream networks, we can visualize the points, extract information about the lake, the points, lights in, and also extract the environmental variables on the points and download as a CSV file. But if we want a more custom workflow, then we can use Hydrographer. For those that join us right now and didn't see the advertisement made by Jaime, so we will be presenting Hydrographer in a workshop next Saturday, so you are invited to join us. Currently, we are using these data sets to model species distribution in the Danube River Basin, for example, in the project Danube for All I mentioned before, but also in another European project, the SSS Water, where the Danube is used as one case study. And in the slide, you can see a screenshot of one of the vignettes of the R package Hydrographer, where one of or a set of the occurrences records of the Danube fish database are used to showcase how to use the Hydrographer. We are about to submit a data paper in scientific data describing this database, and the database will be, after publication, will be also available and could be downloaded as a CSV file from the Freshwater Research and Environment database of our institute, but also could be also accessed through the GeoNode platform, which is the geospatial platform of our institute, also in Berlin. And with that, I want to say thank you on behalf of all the co-authors that contributed to this database.
Thank you, Justiel. Questions for Justiel? No questions. Thank you. Okay, so now we come to the last talk of this session. It's called Open Science Approaches on Assessing Global Aquatic Insect Biodiversity by Aphrodite. Welcome, Aphrodite.
Hello. So I work in the same research group as most of the people in the session. And my PhD, I think it was one of the first guinea pigs that tried to use hydrography 90M and all these high-resolution global data sets to assess the global aquatic insect biodiversity. And I'm saying it was a guinea pig because problems were popping out like mushrooms. So they really highlighted the need for new data structures and the development of new tools that allow scalable hydrographic data processing. Sorry. So freshwater ecosystems cover 1% of the Earth's surface, but they host 10% of its biodiversity. However, until recently, they were assessed and packed together with terrestrial or marine ecosystems in global environmental assessments. But one quarter of freshwater fauna is found to be threatened with extinction, and this really highlights the need for more detailed study and high resolution of freshwater biodiversity at a global scale. So among species that are threatened, specialist species are known to be more threatened. And these are the species that thrive under a narrow range of environmental conditions. So this makes them particularly vulnerable under changing conditions. However, we had a very narrow knowledge on where specialist species of aquatic insects occur in the world. So, as part of my PhD, I worked with aquatic insects that actually are the 64% of the freshwater fauna. And particularly, I worked with Ephemeroptera, Plecoptera, Trichoptera, and Odonata. So these are species that, as adults, they fly out of the river, but they lay their larvae in the river, where the larvae spend a great amount of time of their lifetime. And the larvae of different species have different sensitivities to water quality. That's why they're widely used in water quality assessments. So they are 4 insect orders with global distribution that encompass many, many species across the world. However, 1 out of 3 species is found to be threatened by global change. Despite that, we still don't know where they exactly occur. So there are some challenges in general when we try to map the global aquatic insect biodiversity of any kind of freshwater organism because of certain challenges. First, data is scattered. Second, hydrographic data processing is really challenging, as you saw also from the previous presentations. So this leads to huge knowledge gaps on species distributions. So for this, as you saw from colleagues, we set out to provide scientists with high resolution data, both species data, but also hydrographic and environmental data. And then easy to use, friendly and scalable tools to process this data. So yeah, this gave the name to the study, Open Science Approaches on Assessing Global Aquatic Insect Biodiversity. So for this, I put together 56 datasets from 92 researchers across the world that were hidden in hard disks until then. And then I cleaned all data with some workflows that now might seem obsolete because I saw like today so many wonderful new R packages that really make all these workflows easier. But back then it was a real pain. So after cleaning all the coordinates and standardizing the data, we published the data in the open data repository of our institute, IGB. And I still haven't put the data in GBIF, but today I learned that there is no excuse because there is a package that converts tables to Darwin Core, so I need to do it. We also created interactive maps with Vanessa on GeoNote, so you can zoom in and see what dragonfly is flying next to your home. So the base of these maps was hydrography 9TM, as you saw. And because all these mapping processes and data processing workflows were so complex, we were actually using some open source tools as GDAL and GrassGIS that personally they took me 1.5 years to learn how to use. And then we decided to make them more available and user-friendly, so we wrapped them into an R package called Hydrographer. So at this point, I wanted to tell you that if you want to join the workshop, please check the installation guide beforehand and bring your laptop. Yeah. So then I moved on to map the patterns of aquatic insect biodiversity, and particularly I chose to map patterns of ecological niche breadth, so where the most specialized or less specialized aquatic insect genera occur in the world. So this resulted in maps like this for the 4 orders, where with yellow color you see the more specialized species and with blue the more generalist ones. And then I tried to assess these patterns based on some hypotheses that try to explain the latitudinal gradients. Sorry, about the methods. Yeah, so the special units for the analysis were the subcatchments from the hydrograph in NTM, while I reconstructed the niches using bioclimatic data from GELSA and also topographic variables from the hydrography and soil consistency variables. So I reconstructed the niches using an approach called SENFA, even though now I'm moving to hypervolumes. And then I got values of sensitivity that shows specialization for individual genera, and then I grouped them by assemblage at the subcatchment level and at the climatic biome level. So then I wanted to test this hypothesis. First one is, says that species are more specialized in communities that have a higher species richness. So high species richness, because of resource competition or competition in general, makes them to specialize. However, I didn't find evidence for this hypothesis at the biome level. And then I tested the second one that says that specialization is driven by climate and therefore latitude. So this hypothesis states that in the equator, because of climatic, mainly temperature stability, species are more specialized, while when we move away from the equator, we have a stronger seasonality that makes species to adapt in a greater range of climatic conditions. And for this one, I found slight evidence for the 3 out of the 4 orders. So I am skipping those. Yeah, so I think this work really highlighted the need of scalable tools, because I think it's really impressive that 5 years ago, as I said, there were so many problems, and this led us to make a list of what needs to be done in order to make these workflows reproducible and scalable, and also easy to use for freshwater ecologists. And now we are in a more better state, so I think that this could be done way faster today, and this is hopeful. Yeah, so why this is important? The global EPTO database that I compiled is already being used in other studies of mapping global aquatic insect biodiversity. So now we are able to create species distribution models to predict how species distribution might shift under climate change. And then the tools in Hydrographer help assessing connectivity, which is particularly important. In Europe, at least, we have this EU Green Deal to restore 25 kilometers of free-flowing rivers by 2030. So there's the question what to restore first and which barriers should be removed in order that, so that populations are better connected. So the tools we have created help in this kind of assessments. So closing, I would like to thank my colleagues and thank you.
Questions for Aphrodite? No questions? Okay, so before going for coffee, I just want to highlight what Aphrodite said. You might think that we are computer freaks, but we are actually ecologists, but when we started our research, we find out that there are so many challenges, so many data missing at the proper resolution. So that's why we spend many years getting this data, getting these tools ready. And this is one or the first application where you take all this information to do actually research. So I'm very happy after the first session, we have seen a lot of data and platforms tailored towards researchers, but then you saw also the example of Paula where she has created this platform for interdisciplinarity and has interacting with the stakeholders, so this is also useful. But creating all this data and making this data available has also challenges, and we are going into fair data, but you have seen the challenges, but it was really nice to see how there are protocols and feedbacks to see if you're going in the right direction or not. And finally, you see how this data can be all integrated for research. So this is the end of the first part of this session. The second one is going to be really nice because all the applications for biodiversity conservation, for area prioritization, also for restoration projects, and there is only one person from Germany, so the rest is coming from Australia, Spain, Uganda, and Colombia, so it's going to be really nice. I hope to see you here after the coffee. Thank you.